Now we look at this. The chromosome only for eukaryotic cells. The prokaryotic cells, they don't have chromosome. They're circular DNA. Okay, they don't have the chromosome. So the major carrier of the genetic information in eukaryotes generally is in the form of chromosome, which lie within the cell's nucleus. And in fact, chromosome is a general term we use to describe the DNA material or genetic material inside the nucleus. In fact, at different stages, we give them a special name. Are you clear? Okay, so later I will show you guys. Although this means the chromosome means colored body. Okay, chromosome means colored body. They only give us the color when we stain them. Are you clear? We stain them. Okay, so chromosomes are made out of chromatins. A material consists of DNA and associated histone protein. So what's histone protein? Histone protein, actually, they have positively charged because we do know that DNA is a negatively charged, correct or not? So because the DNA is negatively charged due to the phosphate group, so therefore, they can actually associate with this positively charged histone to form a nucleosome. So it's a fundamental unit of nucleosome consists of bit-like structure with 146 base pair of DNA wrapped around a disc-shaped core of the 8 histone molecule. Another segment of DNA, about 60 nucleotides pair, what we call as a lingual DNA. So look at this, guys. You can see that in this diagram, we have 1, 2, 3, 4, 5, 6, 7, 8. So 8 histone protein actually join together, okay? And you can see our DNA actually wrap around. Okay, so can you see that? Wrap around, then continue to wrap around another histone and continue to form this what we call the nucleosome. So if you look at the DNA, we do know that the diameter of DNA is 2 nanometer. After they wrap onto this histone protein, it form a nucleosome with 10 nanometer diameter. Are you clear? And the parts that are not coiled around this histone protein is termed as the lingual DNA. And this histone protein do have a tail. Can you see that? Do have a tail. This tail is very important. When the tail and tail interaction, it makes these chromatins more compact. Are you clear? Okay. So now we look at this diagram. So first of all, you look at this DNA. You view it under this electron microscope. So DNA is a double helix and the diameter is 2 nanometer. Can you see that? 2 nanometer. So how we organize them? It's something like a thread. We need to organize them, right? So how we organize them? We organize them on a row, okay? A thread row, correct? Okay, we roll on it. Okay, we coil it. So how we coil it? We coil it onto this histone protein. Can you see that? Forming this nucleosome. Okay, so in the between, can you see that? The lighter one, those are the lingual DNA. Okay, lingual DNA, so you can see a coil on it. So if you measure the diameter, now increase to 10 diameter in, okay, 10 nanometer in diameter. And this H1, can you see that this histone and histone, they can actually interact together to make it become a thicker form. So this thicker form is known as the, okay, can you see that the diameter now increased to 30 nanometer. Okay, so if you look at this part, sorry, this part we call as a DNA, this part is DNA, and here we call as the chromatin. Are you clear? So what's chromatin? So DNA plus histone protein, so form the chromatin. Are you clear? And this chromatin, the chromatin condense, okay, to form chromosome. Are you clear? The chromatin condense, okay, to form chromosome. Okay. So it means that if you look at this area here, you can see that this, okay, chromatin start to form loops and then coil onto the protein scaffold, and it form this one sister chromatid. So we have two sister chromatid to form a chromosome. So this chromosome is known as the metaphase chromosome. Sorry, I think I changed the color. So this structure is known as the metaphase chromosome. So you can see that this from the loop, everything, and then coil onto this structure. And this structure, can you see that now increased to 700 nanometer? If a 700 nanometer, generally we can see them under the light microscope, but not the 30 nanometer. 30 nanometer, sorry, we can't. Okay. So 300s may not be able to see, but when they coil into super coil onto this protein scaffold to form a chromosome, then yes, you will be able, or we will be able to see them through by using the light microscope. The rest, we have to use the electron microscope. Clear a lot, guys? Okay, so try to answer the question there. Okay, so the protein known as histone are responsible for the first level DNA packing in the chromatins. Okay, although each histone is very small, it's contains about 100 amino acids, but the total mass of the histone in chromatin roughly equal the mass of DNA because a lot of them, more than a fifth of the histone amino acids, they are positively charged. Eh? So that's why every fifth of the histone amino acid, they are positively charged, and therefore bind tightly to the negatively charged DNA. So we have 4 types of the histone are most common in the chromatin. So just for information only, we have H2A, H2B, H3, and H4. H1 involved in another functions, okay? H2A, H2B, H3, H4. So this is 1, 2, 3, 4. So total, we have 2 of them. And then, so these 4 type histone are critical for the next level of the DNA packing. So in electron microscope unfolded, okay, chromatin is about like 10 nanometer in diameter. Such chromatin resemble beads on a string. So each bead is a nucleosome. Okay, so we have seen this right just now. Can you see that? The beads on a string. Okay. So the string between the bead is known as the linker DNA. So a nucleosome consists of DNA bound twice around a protein core of 8 histone, 2 each of the main types, H2A, H2B, H3, and H4. The amino end of each histone, they have the tail extended outward from a nucleosome. So in the cell cycle, the histone leave the DNA only briefly during DNA replication and transcription. So try to imagine, guys, if this is our histone protein, so our DNA actually wrap around it. Okay, can I see that? So if a DNA wrap around it, can I do the transcription? Cannot. Can I do the DNA replication? Cannot. So it means that only during the DNA replications and also transcription, then this histone protein actually away from the DNA, so that DNA can perform the transcriptions and also the DNA replication. Okay. So the next level of packing results from the interaction between the histone tail of one nucleosome and the linker DNA and nucleosome on either side. So the 5th histone, so which is H1, involved at this level. So make them even packed more tight together. So this interaction caused extended 10 nanometer fiber to coil or fold, forming a chromatin roughly about 30 nanometer thickness, okay, or known as 30 nanometer fiber. And this fiber then form the loops, okay, called the loops domain attached to a chromosome scaffold. Scaffold means that it's like skeleton, okay? So we have something like skeleton around these chromatins to coil onto it. So then because of this, they form about 300 nanometer fiber. So in a mitotic chromosome, the loop domain themselves coil and fold in a manner not yet fully understood, further compacting all the chromatin to produce the characteristics of a metaphase chromosome, metaphase chromosome. So the width of a chromatin is about 700 nanometer. Particular gene always end up located at the same locus in metaphase chromosome, indicating that the packing that are highly specific and precise. Now, this is very, very important. When the packaging takes place, it always have the same locations of the gene. So later, we're going to look at this, the terminology in terms of the locus, okay? Locus and also some other terminology. So during interphase, we have two types of the chromatins, when the euchromatins and also the heterochromatin. Euchromatin, can you see that are loosely packed? Heterochromatins, they are tightly packed. So this is the nucleolus. Okay. So in the early stage of mitosis or meiosis, or we term as nuclear divisions, the chromatin double helix become more and more condensed. Okay, so it become more and more visible. This form of the classic four-arm structure is known as metaphase chromosome, which have a pair of the sister chromatid attached to each at the centromere. So you can see that, guys. This is one sister chromatid, another sister chromatid. Okay, so you can see that one sister chromatid, another one sister chromatid, they're identical. So they coil, and then we have someone to actually hold at the center part. Even though we call center, not necessarily must be center. Okay. So the short arm are called, because of the position of centromere, you can see that we have short arm and long arm. So short arm is called P called petite, petite means small, and the longer arm called Q arm. See? Can you see that? Okay, long arm. Okay. So we have the short arm. Now, each individual of a given species have a characteristic number of chromosomes in the nuclei. So, we have two sets, so 22 in total. Autosome. Autosome means that nothing to do with the sex. Chromosome, determination of the sex or gender, and two sex chromosomes. So total we have 46. Now we understand the haploid and diploid because somatic or normal cell will contain two sets of chromosomes. So therefore, it's termed as diploid. Okay, so this is represented by 2n, where n is the number of chromosomes in one set of chromosomes. But not all our cells, they are diploid. Some of the cells, they are haploid, only one set, which are gamete, so represented by n. So these 2n cells actually can get back n through the process of meiosis, okay, or reduction division. Now, if you look at this DNA, the chromosome, we put it side by side, you'll realize that a gene locus is the positions of the particular gene on a chromosome. So they always have the same locus. Okay, so it depends on the size of the chromosome. The longer the chromosome is, or the bigger the chromosome is, the more genes they have. For example, chromosome 1, they have about 2,000 gene loci. Chromosome Y, the smallest number, we have about 50 gene loci. Okay. So what is locus? Locus basically address or positions of a particular gene. So it won't change, okay? It won't change at all. For example, this is chromosome number 7. So this chromosome number 7, okay, this chromosome number 7, you can see that you have this. Interference beta 2 at the P arm, we have the PSP, the EGFR, EPO, CF, T cell receptor, okay? But you can see that, guys, they have their own specific location. So it means that if I want to find EPO, you in your cells and my cells, all human cells, we're going to look at the Q arm of chromosome 7. You don't go to the chromosome 8 and find them. You won't be able to find them because the address is not there. Are you clear? So there's something like condominium. You can see there's something like condominium or apartments. So each floor has a particular gene to be there. So that floor we call as a locus. Are you clear? Okay. So I'm going to summarize here. Now, what's mean karyotype? Karyotype basically is a number and appearance of chromosome in the nucleus of a eukaryotic cells. So karyotype describes the chromosome count of an organism and what this chromosome look like under the light microscope. So we pay attention to the length, any change in the length, the position of the centromere, and whether they are too near to the top part or too near to the bottom part. Bending pattern when we dye it or stain them, or any difference between the sex chromosome. Okay, we call it XX or XY, for example, for human. So the preparations and the study of karyotype is a part of cytogenetics. Cyto means cells, genetics, so it's a cell genetics, okay? So karyotype is prepared by cutting out individual chromosomes from a picture and rearrange them into the karyogram. So homologous chromosomes are arranged in pair. Autosome pair are arranged in descending order according to the length, and sex chromosome display at one side. So that easily for us to determine this sample belongs to the male or sample belongs to the female. So how we do this, guys? So we have the slide. So we drop the sample. When we drop the sample, then we can stain the sample. When we stain the sample already, we can take a photo. So when I take a photo, what we have here, can you see that, guys, in this diagram? So you can see that it's the color. Can you see that very, very precise no color? So we can actually cut out. So you see that we cut out and put them together. So this is a karyotype. And sex chromosome, we always put it on one side. Can you see that? Easier for us to identify, oh, this is XY, so it must be a male sample. Can you see that? Okay. And then you can see that this when how we arrange it into the descending order in terms of the length. Okay, the longer one first, the shorter one at the last part. Okay. So karyotype can be used to study the chromosomal abbreviations. So what means is mutation, chromosomal mutation. Okay, so we can use these karyotypings to help us to study the chromosomal mutation. So we can check in terms of number or in terms of structure. For example, we do have the Turner syndrome, single X chromosome. So total chromosome 45. So X chromosome only, so 45 XO. So Turner syndrome. We have a Klinefelter syndrome, the most common male chromosomal disease, otherwise known as 47XXY, is caused by an extra X chromosome. So because extra X chromosome, so this Klinefelter is male, but they do have the characteristic of female. Right, Klinefelter. We have Down syndrome, where we have trisomy of chromosome 21, and we have this cri du chat, okay, or in France, I don't know how to pronounce this, but basically it's a cry of the cat when the truncated short arm on the chromosome 5 cause the voice box of the individual change. So when this person cry, it sounds like a cat crying. Okay, because of the mutation. So you can see the guys in this diagram, if I enlarge it, so A actually trisomy 21, Down syndrome. Can I see that? Down syndrome. So we can actually arrange A. How come we have extra chromosome 21? So it means that it must be Down syndrome. Okay. Now, how about B? What is the syndrome? What is the disease? What's the disease? Can you see that only X chromosome without Y chromosome, or only one single? So in this 45 only, so this is a Turner syndrome. XO, Turner syndrome. If you look at this, we have double X and Y, double X and Y. So C actually refer to the Klinefelter, Klinefelter syndrome. And then the last one, can you see that, okay, in terms of the number, they are the same, but can you see that one chromosome at the chromosome 5 becomes shorter already? So this is cri du chat. So again, cri du chat, I mean a syndrome where the mutation caused the change in the voice box, results in the change in the sound. Okay. So with this, I have done for these the topics of karyotypes of the recording.